Good afternoon, everyone, and thank you for coming to listening my presentation on structural insight into TRPV channels gating. And of course, thank you to Beckman Foundation for inviting me to this symposium to give this presentation. Transient receptor potentials are non-selective cation channels that are involved in many physiological and pathophysiological processes. They're composed of 28 homologs, and my lab in the last almost 15 years has been studying the subfamily of those channels called TRPVs, and specifically we're interested in the channel TRPV2. And the reason for it is that this channel actually has a very interesting physiological function, and it's expressed everywhere in the body. And it's non-selective calcium channel and has been shown by numerous studies involved in very important physiological processes and also involved in human disease. Nevertheless, very little is understood about this channel at the molecular level. We, in 2016, was able to solve a structure of this channel and sort of present the first topological representation of this channel. So it's a tetramer, as you could see here. It's one monomer composed of six transmembrane domains. It has N and C termini inside of the cell. And on the N terminus, it contains 6 anchoring repeats, which you could see here. And C terminus has a TRP helix and STD domain that represents as a beta sheet. So TRPV also has a lot of activators, but they all non-specific. So we, of course, to try to understand this protein at the molecular level and their physiological function, would like to utilize this non-specific activators and some inhibitors to find the binding sites to further design of much more specific compounds that would activate or inhibit the channel. In this case, that I'm only presenting, we focused on 2 compounds. One is the 2-ABP, is the activator of the TRPV2, and ruthenium red, what is the pore blocker of the channel. So we employed cryo-electron microscopy and tried to answer the question, if using that technology, can we discover the whole gating cycle of the channel upon activation by one of those compounds? In this case, we will be using 2-APV. And so the first thing we had to solve using cryo-electron microscopy, the overall apo structure of the channel at the higher resolution. And to our surprise, we actually discovered that the channel could contain 2 sorts of substate in apo state. It has apo1 state and apo2 state. They are basically a closed channel, so no ion could go through the pore of the channel. This is the representation of the pore of the channels. But interestingly, the selectivity filter of one of the states, APO1, was tightly closed, while the selectivity filter of the APO2 state was widely open. Nevertheless, the lower gate, that is at the methionine 645, was closed in the APO1 and APO2 states. This actually was quite interesting finding. So next we decide to see how is the compound that we would like to study affect the opening of the channel. So we know that 2-ABP could open the channel, and of course the channel undergoes opening and closing conformations pretty fast. So we apply 2-ABP on our cryo-electron microscopy grids and solve the structure in the presence of 2-ABP. We actually had to utilize CryoSpark software which allowed us to actually sort in real time almost to see several substates of this channel or the transition of the channel from open to closed state. What was also more exciting that at the sort of both minimum of this opening and closing, we were able to solve 2 structures, one of which was what we called in TRPV2 2-ABP activated state, so more open state, and TRPV2 inactivated state that would be much more closed or basically undergoing pore closure. So we also were able to identify where the 2-ABP binds, and we found that 2-ABP actually binds in a pocket closer to the intracellular membrane part of the channel, and it binds between 2 monomers of the tetramer, and it basically interacts with the arginine, tyrosine, threonine, and histidine in a pocket that is located between S6, S5 of one subunit, as I mentioned, and S4, S5 of another. And you could see here, this is the apostructure. And in apostructure, this pocket is actually quite tightly shut. And where in the 2-ABP part, the drug actually has to wedge itself into this pocket to open the channel. Of course, to confirm that this is the binding site, we did electrophysiology, and we basically mutate all these residues to alanine and was able to show that actually histidine play important role in the interaction of the 2-ABP with the channel in this pocket, while other amino acids allowing this for the coordination. So next, we looked at the overall pore of the channel, and as you already could predict, the apo state, as I said, was closed. The 2-ABP activated state showed us a much wider open pore, and inactivated state, what would be sort of going back to the closure, sort of was in between. And interestingly, also, this histidine on position 651, located between TRP helix and S6, was actually going from rotating, basically, as you could see here, from inside of the pore to outside of the pore. And this is just the representation, and sort of forming this almost a connection with the S4, S5 linker and holding channel into the open conformation. And I guess this interaction sort of gets relaxed, and when the protein goes back to the more closed state. So what also interesting, we noticed that this C-terminus conformation of the channel at the apo state and in the activated states, it sort of is in alpha helical-like conformation. But when it goes into the inactivation, it sort of forms like this unstructured loop that goes and interact with one of the anchoring repeats in the other subunit. And that has been seen before for several other trip channels, suggesting that this basically N to C terminal switch is important for channel activation and activation, and we basically see this in our structure. So now, what is the conformation changes the channel undergoes when it's ceased to ABP and opens? So it's a large rotation for the anchoring repeats, almost 7-degree counterclockwise, and to ABP actually by wedging in that pocket basically moves the S4, S1 bundle helix away from the center axis pore, almost again 7 degrees, and also pushes the S4, S5 linker up together with the obviously S6 and pore helix up, and so that's sort of widening the pore. And on top of it, if we look from the side, you clearly see that the channel overall by itself is sort of widening with all these conformations. So we could say that by looking at our gating cycle, we sort of was able to resolve conformation changes, how the channel undergoes from apo towards activated state. Next, we obviously compare the structures of activated state to the other structure, that what we call inactivated state, and we notice absolutely almost like a reverse mechanism is that the anchoring repeats now moving 7 degrees counterclockwise, and that brings back the S4, S1 helical bundle back, and it's sort of, the drug is still there, so the binding pocket is still slightly widened, of course, because the drug didn't left yet the protein, but some other conformations sort of making that the S5 and S6 and P helix sort of again goes down and sort of making the channel, as you could see here, sort of going backward, like not widen, now shrink. And more interestingly, all of those conformations lead to sort of motion of bringing anchoring repeat domains down almost by 4 angstrom. So this is an interesting conformation that now we could say that we see the activated state transitioning towards the inactivated state, and eventually it will transition to the apo state. So here is the movie to show all this conformation. So you could see here, that's the widening and opening of the pore. And now if the channel gets inactivated, we see the channel sort of going back to the closed position. And if you haven't seen it, if you look on the bottom, this alpha helix on the C terminus is basically transition from alpha helical conformation to sort of this loop-like conformation. And channel goes into inactivated desensitized state. So we also looked at the ruthenium red, the compound that actually binds inside of the pore of the channel and blocks it. So we know that it's a very potent blocker. And so what we did next, we solved the structure of the channel in the presence of ruthenium red. We were able, again, resolve not just one conformation, but actually two, one of which we called ruthenium red 1 and another one we called ruthenium 2. And this one represents absolutely a common apostate. And here we saw, as you could maybe already see here, so some density inside of the pore that we were able to assign for ruthenium red. And here is the atomic representation of how we think ruthenium red possibly interacts with the pore of the channel. So it actually goes inside of the pore because it's widened in this ruthenium 2 state, similar to the APO2 state that I showed earlier, and basically blocks the pore, presenting basically a very tight block here in the selectivity filter, resembling almost in the present of the ruthenium red. Here is into the brown color. It's completely a blocked channel and none of the ions could go through. So of course, to confirm that, we did mutagenesis of this residue that we think interacts with the ruthenium red. It's glutamic acid 609. And we could see that abolishing basically this E609 actually prevents the block of the channel. So based on all our recent studies, we could say that we were able to resolve, using cryo-electron microscopy, a cycle of activation and inactivation of the channel, in addition to basically a pore block of the channel. So together from all our studies, our bigger conclusion is that selectivity filter of TRPV2 is not a gate as been proposed before, but instead it has a conformational flexibility allowing entry of large positive charge pore blocker like ruthenium red. The binding of 2-ABP and interface between S4, S5 linker on one monomer and S5 on adjusted monomer leads to jet channel opening, subsequent inactivation. Glutamic acid 609 is a key residue to determine the ruthenium red block, and histidine 521 locates over here is a key residue for 2-ABP binding. And of course, it wouldn't be possible. All these experiments were done by two outstanding senior research staff members, and Anna Protopopova and Ruth Pomeroy, with the help of other members of the lab, in collaboration with Andreas Leffler Lab and Hanover Medical School and Vladimir Yarovo and UC Davis. And of course, the NIGMS and Beckman Foundation sponsored these studies, and we would not be able to do all our cryo-electron microscopy if we were not funded by Beckman Foundation for establishing of our cryo-electron microscopy center. Thank you very much for your attention, and I will be taking questions. Thank you.